All right, so we covered prokaryotic gene expression regulation. And so prokaryotic gene expression regulation is going to be a lot simpler than eukaryotic gene expression regulation, but they're going to follow a lot of similar rules. So we're going to express these genes in response to environmental cues or not expressed in the response to environmental cues. And so this is the way that cells can conserve resources. So what we talked about with prokaryotic gene expression regulation was mainly this level of transcriptional control. So we control whether or not we're going to be transcribing RNA for that gene. But when we talk about eukaryotic gene expression regulation, there's going to be more levels that we can use to regulate the gene expression. So it's usually the case that when we look at eukaryotic cells, we see kind of more complex behavior with like more levels to it. And gene expression is not an exception to this. So the first level, kind of starting with the DNA, so if we look at it from the DNA going out to the proteins, if we start with the DNA first, just the way that our genes get packaged affects how well they get expressed or how often they get expressed. So we have our genes, our DNA is wrapped around these proteins called histones, and then all gets packed up together, and that's called chromatin. So chromatin is this mixture of DNA and proteins. And so if DNA is wrapped really tightly around these histones and kind of hidden from view, that means our RNA polymerase can't get in there and transcribe it, so it's not going to get expressed. So just the way that our DNA gets wrapped around these histones influences how often it gets expressed. So that's one level of gene expression regulation. Then we have transcriptional control, which is going to determine how well an RNA polymerase can bind to our DNA to make mRNA. So that's like what we talked about with prokaryotic gene expression regulation. We have post-transcriptional control. So remember we talked about like alternative mRNA splicing, how we cut out different introns for different types of gene expression. So after we've made our mRNA transcript with our RNA polymerase, we can process it in different ways. And this is going to also determine how much protein that we make. Then the next level, we have translational control. So how the protein gets translated from this mRNA, the cell can also control that. And so that's going to determine how much active protein we have out there in any one time. And then finally, we have this post-translational control. And this is after we've made these proteins, they all get broken down over time. And so if it doesn't get broken down for a long time, we would consider that gene to still be expressed. So if we have a gene that makes an enzyme that maybe breaks down lactose, and the way we made that protein means that it's never going to get broken down, then that would mean that gene is expressed for a very long time because its protein is still out there doing things. If we made a protein that's going to get broken down like right away, then right after it gets broken down, we would say that gene is not being expressed any longer. So that's kind of like an overview of what the different levels of gene expression regulation will be in eukaryotic cell. Now we're going to go into a little more detail for each one. So again, we talked a little bit about this idea of chromatin. This is like DNA that's wrapped around proteins. It's sort of like if you're organized, when you store your Christmas lights, you maybe like wrap them up in a spool before you put them into a box, or you can just like jam them into a box like what I do. But if things are like DNA is wrapped really tightly around a histone and like hidden from view, then RNA polymerase can't get in there. So tightly packed DNA cannot be transcribed. Loosely packed DNA can be readily transcribed. So the cell can wrap the DNA differently based on different environmental cues. We're not really going to talk about how the cell can respond to those cues, but it's similar to prokaryotic gene expression regulation, where there's some change in the environment that changes some of the binding properties of these different proteins or biomolecules and influences how they're wrapped around this histone. So this is this first level. We're still in the DNA, and just the way we package it determines gene expression. Any questions about that concept? No? Okay. Transcriptional control. This is similar to what we see with prokaryotic gene expression regulation, but it can be a little more complex because the promoter doesn't always have to be near these other kind of regulatory units. So here we have an enhancer, which is sort of like an operator. It's similar, but just in a different way. But what's relevant here is that with eukaryotic gene expression regulation, we can have DNA that's far away down the chromosome come back and loop over and influence transcription. But it's really like the same operand model that we saw in prokaryotic gene expression regulation. The important part to this level of gene expression regulation is that we can influence how well RNA polymerase binds to the DNA, and that's going to influence how these proteins get expressed, just like in prokaryotic cells. So, post-transcriptional control. So we've talked about this idea of alternative mRNA splicing a little bit in the past. So we have a transcript, or here they call it a pre-mRNA, and what regions we cut out as introns. So here we cut out these sections as introns. Here we cut out this section C as an intron. So we can produce two very different proteins, protein 1 or protein 2, based on how we cut out different regions as introns during our mRNA splicing phase. And we can change the mRNA splicing based on different chemical environments. So what regions of the DNA, or what regions of the RNA that get cut out as introns can be controlled by the cell in a way that's sensitive to the chemical environment to allow it to produce different proteins when it needs to. Introns? Yeah. Introns? What about, sorry.
I thought that was a question.
Oh, yeah, no, you're, okay, I'm sorry. Yeah, I was saying like what when introns get cut out can be influenced by the chemical environment. Sorry, it was like a half rhetorical, half actual question. My bad. Sorry. But what's important here is that this is controlled again by the chemical environment. So the point of this, going through all these levels is to show that there's a lot of ways that the cell can regulate its gene expression, and they're all controlled by their environment. So it allows the cell to respond and adapt to changes in the environment. And these changes in the environment can be like the change in food sources that are presented, but they can also be the change in like what hormones the body is releasing, or things like that, like signaling from other cells. So cells can signal other cells to change their behavior. And this is the way that big multicellular organisms can do a lot of like complex tasks. So there's two more layers left. This translational control. So after we have this RNA transcript, we're going to add on a 5' cap and a poly A tail. And how long this poly A tail is going to determine how long this RNA is going to stay intact and be able to be used for translation. So remember, what's going to happen for translation is the ribosome is going to come, it's going to attach to this RNA, and it's going to start making proteins based on these instructions. But if on the end of our RNA we have a very short poly A tail, we're going to have enzymes that come along and just kind of like eat it from the end, like a Pac-Man, and break it down. So we have a really short tail. This is going to get broken down really quickly, and then the ribosome can't attach to it and make proteins. If we give this a really long poly A tail, it's going to take a long time for the enzymes to break that down, and this is going to be floating around for a very long time, and we're going to make a lot of proteins from it. So the cell can decide how long it wants to be making proteins based on how long it makes this poly A tail or how well it puts on this 5' cap. So does everyone kind of understand the concept of how the stability of the mRNA influences gene expression? So gene expression is the actual making the proteins. So if we stop the process at an intermediate step, like if we break down this RNA at the end of transcription, then we're not going to be able to translate that gene into proteins because we won't have the RNA to do it.
Does translation always include splicing?
Yeah, so that's really more of like one of the steps at the end of transcription, but it always includes the splicing step. So the final step the final level of gene expression regulation in eukaryotic cells is this post-translational control. So after we make a protein, you can add a tag onto it that helps the cell determine, how quickly to break it down. So there's these enzymes that break down proteins, and if they have this tag on them, they can get broken down faster or slower, depending on what kind of tag it is and what the cell wants to do. But if the protein is made and it's tagged and it gets degraded really quickly, then we're not going to have an active protein anymore. And so we're no longer expressing that gene. So even after we went through all of the energy of making the protein from that gene, so we went to the DNA, we made an RNA transcript, we processed it, we sent it out to the cell, we made our protein, we can then tag that protein and say, okay, I want this gone in like X amount of time, or some amount of time. That'll determine how quickly the proteasome, the enzyme that comes to the protein, comes along and breaks it down. So the cell can control this whole process by tagging the protein in different ways. And if it breaks it down, we will consider that gene to be no longer expressed. If it doesn't break it down, that gene is still expressed. All right, that's the information to answer the last 5 questions on the review sheet. Does anyone have any?
So for the review that you have in these slides, these quick questions here.
Yeah.
Those are what we should just focus on, like the alternative mRNA splicing, and then the knowing the poly-A2 only, rather than what we translated.
Yeah, these are, if you should be able to answer those questions as well. Yeah. So yeah, if you have the time, try to answer these questions, and you can email me, or I can post the answers to those questions on the end of this answer key as well. But you should go through those and try to figure them out for yourself. And also just briefly define what the different layers of eukaryotic gene expression regulation are on the back of the sheet. And I'll post an answer key for all this.
Are we still having a review today?
Yeah.
Are any foods or anything come with enzymes? Do you know any?
Yeah, but usually those would get broken down in your stomach before they enter your bloodstream. But yeah, I mean, all your food is packed with enzymes because they're in the cells of the animals that you, well, the plants or animals that you eat. But yeah, your stomach is very acidic, and your digestive system is very good at breaking down functional biomolecules. So they don't really get into your.
Would that be any different from eating like just protein or amino acids? Like it's all just going to get broken down and put back together.
Yeah, I don't know what, maybe like retains some. Yeah, I wonder if you could eat all of your food just pulverized into like individual biomolecules. I don't know how that would be different.
I don't know, I was watching a show, and somebody in the show had read an article about some food that had enzymes in it. I was wondering if that was words.
Yeah. I mean, yeah, it all has active enzymes. Okay, that's what I wanted to cover for today. I'll be around for 5. I'm sorry. I'll be around. Thank you.